Here are 3 high-yield USMLE-style questions focused on enzyme terminology. 1. A 45-year-old male presents with fatigue and muscle weakness. Laboratory tests show elevated serum creatine kinase levels. A genetic analysis reveals a mutation in the gene encoding glycogen phosphorylase. Which of the following best describes the function of glycogen phosphorylase? A. Transfers phosphate group from ATP to glucose. B. Transfers a phosphate group from inorganic phosphate to glucose. C. Removes a phosphate group from glucose. D. Adds a phosphate group to glucose using ATP. Answer B. Transfers a phosphate group from inorganic phosphate to glucose. Explanation: glycogen phosphorylase is an enzyme that adds an inorganic phosphate onto glucose residues in glycogen without using ATP, breaking down glycogen into glucose 1-phosphate. This process is different from kinases, which use ATP to transfer phosphate groups. 2. A 60-year-old woman is diagnosed with a metabolic disorder involving reduced glucose production. A laboratory study reveals a deficiency in fructose 1,6-bisphosphatase. Which of the following enzymatic functions does fructose 1,6-bisphosphatase perform? A. It adds a phosphate group to fructose 1,6-bisphosphate. B. It removes a phosphate group from fructose 1,6-bisphosphate. C. It transfers a phosphate group from ATP to fructose. D. It adds an inorganic phosphate to glucose 6-phosphate. Answer B. It removes a phosphate group from fructose 1,6-bisphosphate. Explanation: fructose 1,6-bisphosphatase is a phosphatase enzyme that catalyzes the removal of a phosphate group from fructose 1,6-bisphosphate, converting it to fructose 6-phosphate during gluconeogenesis. This is the opposite of the action of kinases. Tanner 3. A 25-year-old male presents with muscle cramps after exercise. Lab results show elevated blood glucose levels. Genetic testing reveals a mutation in the gene encoding phosphofructokinase. Which of the following is most likely the role of phosphofructokinase? A. Phosphorylates glucose to glucose 6-phosphate. B. Adds an inorganic phosphate to fructose 6-phosphate. C. Transfers a phosphate group from ATP to fructose 6-phosphate. D. Removes a phosphate group from fructose 1,6-bisphosphate. Answer: C. Transfers a phosphate group from ATP to fructose 6-phosphate. Explanation: phosphofructokinase is a kinase that catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by transferring a phosphate group from ATP to fructose 6-phosphate, an essential step in the glycolysis pathway. These questions are